Good afternoon, students. We are starting zoology class now.
Good afternoon to everybody. In the last class, you studied about the Griffith experiment. That means which one is the genetic material, whether it is a protein or a DNA is a genetic material. So today we are going to discuss about DNA is the genetic material. So in that one, many biologists like Griffith, Array and others still believe that proteins were the genetic material. That proteins were the genetic material. DNA was not genetic material. You understand? Many of the biologists like Griffith, Array and others still believing that is a protein were the genetic material. DNA was not genetic material. In that condition, in 1952, Hershey and Chase conducted an experiment to prove DNA is the genetic material. They want to prove that DNA only genetic material, not protein. For that purpose, in 1952, Hershey and Chase conducted an experiment to prove that DNA is the genetic material. They conducted an experiment on bacteriophage. So what is bacteriophage? So the diagram being given in your book. So see this diagram. This one is bacteriophage diagram. T2 bacteriophage. The bacteriophage is called infected bacteria. The infected bacteria. The bacteria is infected by the virus. That one is known as the bacteriophage. This bacteriophage has a three part: collar region, head region, collar region, and tail region. The tail region having the fibrous. This part only attaching on the surface of the bacteria. This region only known as the basal plate. This region is called the basal plate. This page, that means T2 page is here, virus. See, listen, bacteriophage means infected bacteria. The bacteria is infected by the virus. So the page T2 bacteriophage is here, virus. After the infection of the virus, the bacteria is called the T2 bacteriophage. So this infecting there, what type of the bacteria? That is called the E. coli bacteria. Escherichia coli is called, shortly called the E. coli. When page, that means page means virus. Page means virus. When the virus are added to the bacteria, they observe the outer surface of the some material. Outer surface. This one is the bacteria. Looking like that. This is the one of the virus. The virus is affecting on the surface of the bacteria. They observe the outer surface. The virus observe the outer surface of the bacteria, and some material only enter the inside of the bacteria. Some materials are enter the inside of the bacteria. So then the bacteria is lyses. Lyses means so that destroyed and release the number of the progeny pages. Pages means virus. You understand? Bacteriophage means after the infection of the virus, the bacteria is, we are saying the bacteriophage. The bacteriophage means that is a virus. The virus infecting the fresh bacteria. So something is entering from the virus to the bacteria. That something only we don't know whether it is a protein or otherwise a DNA. There only is somewhat confusion. So what material is entered from the virus to the bacteria? That only discovered, proved with the help of the experiment. That experiment is known as the Hershey-Chase experiment. That Hershey-Chase experiment was conducted by the Hershey and Chase. The pronunciation Hershey and Chase experiment. This experiment conducted in 1952. Now we are going to see about the Hershey-Chase experiment. See, in the Hershey and Chase experiment, this experiment we are taking is they wanted to observe whether the DNA or protein enter the bacteria, whether the DNA or protein enter the bacteria. That was the main concept of this experiment. Which type of the substance entered from the virus to the bacteria and lyses the bacteria and form the number of the progeny. That only the basic concept of this experiment. So they so found into what whether the DNA or protein entered the bacteria. So they conducted number of the or series of the experiment. They conduct the series of the experiment. This experiment takes place by 3 steps. The first step is that the experiment consists of labeling a group of T2 pages. You know what is pages? The virus what we are saying the pages. The T2 pages was with radioactive sulfur 35. The radioactive sulfur is what? Yes, 35. By growing them in E. coli bacteria, labeled with S35. See in the diagram. This one is S35 labeled bacteria. The sulfur is component of the protein. The sulfur is the component of the protein. Sulfur is the component of the protein and not a DNA. Only it is a component of the protein, not DNA. Hence, this page is incorporate S35 in the protein code. So it has a S35 in the protein code. Do you understand the first step? There is an experiment on considered. That is a labeling a group of T2 pages with radioactivity sulfur S35. The second step, the experiment consists of labeling a group of T2 pages with radioactivity phosphorus 32. Phosphorus 32 by growing them in E. coli. Growing them in the E. coli bacteria. Labeled with P32. That is phosphorus 32. The phosphorus is the component of DNA. Phosphorus is a component of the DNA, not a protein. Hence, these pages incorporated P32 in the DNA. You understand? First, they are taking one separate, there is a 2 type of the bacteria and a 2 type of the pages. One pages are living in the medium, that is S35. Another one virus is living in the medium phosphorus 32. Now, both pages are infected the bacteria. That is a first step infection. Okay, so after the infection, something is processed from the virus body to the inside of the bacteria. Virus body to the inside of the bacteria. That condition is known as that step is known as the blending step. That is called the blending step. Okay, these labeled pages are allowed to infect to 2 separate E. coli. Soon after the infection, before lysing means before death. So the bacterial cells are education. The key word is the education. They are making shaking. That part is shaking. That means agitation a blender. P32 was bounded associated with the bacterial cells. 30P32 is present the inside of the bacterial cells, and S35 is present in the outside of the bacterial cells. Outside of the bacterial cells. We can keep this one in that is a test tube also. Like that, we can take the test tube and we are putting everything in the inside and making the centrifugation, making the centrifugation, so that then the supernatant part will come and that is a basic part will come. So the base region, all the DNA particles are present, and the supernatant region, only the protein part will present. We understand? First, they are selecting the 2 type of the pages. One page is living in the ES35, that means radioactivity substance, sulfur 35, and another one page is living in the phosphorus 32, that is a isotope. So now the 2 pages also inserted into introduced into the 2 bacteria. The 2 bacteria, that is called making the infection to the bacteria. After the infection, these labeled pages are allowed to enter the bacteria. Before lysis, the bacterial cells are making agitation and blender. So the P30 was found in the inside of the bacterial cell. And sulfur, that means S35 radioactivity substances are present in the surrounding of the cells, surrounding of the medium or surrounding of the cell. After the radioactivity test, we can see the progeny, that is progeny is small ones, young ones, pages contain phosphorus in the inside of the cell and phosphorus inside of the cell and sulfur in the outside of the cell. Sulfur in the outside of the cell. They cleared DNA only carries the hereditary information from the virus to the inside of the bacterial cell. Do you understand? So sulfur is detecting only the supernatant. Suppose we are taking like this meals in the test tube we are making. So in the inside of the test tube we are sending these things. The supernatant part we can see the sulfur, and so phosphorus we can't see in the supernatant part. It will filter in the below the region. So this is a Hershey and that is a Hershey and Chase experiment. It is otherwise called the transduction experiment. Transduction experiment, or otherwise known as the blending experiment, or otherwise we called it is unmistakable experiment. Because from this experiment only, the scientists are explained only the protein, that is a DNA material only can pass from the virus to the bacteria. The sulfur material so does not enter in the inside of the cell because it always present only in the port of the bacterial cell. It can present only in the port of the bacterial cell. It does not move in inside of the bacterial cell. So only DNA only can move in the inside of the bacterial cell because the DNA only transmitting material, that is transforming the genetic condition or genetic things from one organism to the another organism. So from this experiment, we can understand that DNA is the genetic material, not protein. That is confirmed by the help of the Hershey and Chase experiment. In the last class, we studied about the drift experiment. In that experiment, the scientists didn't come for the particular conclusion. Definitely the DNA only the genetic material, like that they did get the conclusion. But after the experiment of the Hershey and Chase experiment, so all the scientists are understood as well as the concluded the genetic material is only the DNA. Do you understand? What is the other name for the Hershey and Chase experiment? Transduction experiment, or otherwise known as the blending experiment, because this experiment takes place by 3 steps. First step is the infection, second step is the blending, and third step is the centrifugation. So infection means the virus are infecting the bacteria. That means enter in the inside of the bacteria. That is called the infection. After the entering, so the virus are sending some particles inside of the bacteria. So that is known as the blending. You understand? First touching on the surface of the bacteria, that is known as the infection. After the infection, some substance are so moving from the virus body to the inside of the bacteria. So that is called the blending. So after the blending, so that is fully infecting the bacteria before lysis of the bacteria, that is making the centrifugation, that means agitation. From that agitation, so the particles, the DNA particles only present the inside of the bacteria, and the sulfur radioactivity substance are present in the only outside of the port of bacteria. It does not enter the inside of the cell. From this experiment, we can understand sulfur does not present in the inside of the cell, that is in the nucleic acid, and only the DNA only present in the inside of the cell. So we can conclude so that DNA only the genetic material. Okay, so next one. So chemistry of the nucleic acid. Next one is called the chemistry of the nucleic acid. So in the chemistry of the nucleic acid, the genetic material as the nucleic acid has 2 types. One is the DNA, another one is the RNA. One is the DNA and another one is the RNA. You know what is nucleic acid? The nucleic acid which is present in the inside of the nucleus. So it is called the nucleic acid. This nucleic acid are genetic material. So the nucleic acids are genetic material. There is 2 types of the nucleic acids are present in the organism. One is DNA, another one is the RNA. These nucleic acids area long chain. It is a long chain and are polymer of the repeating subunits. Repeating subunits, that is called the nucleotides. That is known as nucleotide. They will ask the question, what is nucleotide? Nucleic acids area making long chain or polymer of the repeating subunit that is called the nucleotides. Okay. So what are the parts present in the nucleotides? One is the pentose sugar, second one is the nitrogenous bases, and third one is the phosphate group. So these three components are present in the nucleotides. First one pentose sugar, second one nitrogenous bases, and third one phosphate. Okay. First, we want to know about the what is pentose sugar. Depending on the pentose sugar, that is sugar, pentose sugar. Pentose is 5. The pentose sugar, there are two types of the nucleic acid. One is DNA, another one is the RNA. See in the structure. So the structure didn't give me a book. So see the deoxyribose, only known as the DNA. Ribose sugar, only known as the RNA. See that depending on the pentose sugar, how the two types of the nucleic acids are classified. The nucleic acids are classified based on the pentose sugar into two types. One is DNA and the other one is the RNA. The DNA is present in the nucleus of eukaryotes. DNA is present in the nucleus of the eukaryotes. So what is eukaryotes? The cells which are present in the multicellular organisms, that is called the eukaryotic cells. That means well-developed nucleus is present. Eukaryotes means well-developed nucleus is present. Well-developed nucleus. The RNA is present in the nuclei of prokaryotes. Prokaryotes means not well-developed nucleus is present. Not well-developed. The cells which has a not well-developed nucleus, that organisms are known as the prokaryotes. Eukaryotes means well-developed nucleus is present. Differentiate between the eukaryotes and prokaryotes. In the first class, we studied that one. So what type of the nucleic acid is present in the eukaryotes? DNA is present in the nucleus, that are seen. DNA and RNA, both nucleic acids present in our body. So in the inside of the nucleus, what type of the nucleic acid is present in the eukaryotes? Eukaryotes as the cell as a nucleus. It is a cell. So it is cytoplasm. It is nucleus. In the eukaryote cells, so what type of the nucleic acid is present in the nucleus? Only the DNA. Suppose we are taking the prokaryotes. In the type of cell, the inside of the nucleus as a nucleic acid is the RNA. So what is difference between these two nucleic acids? Only one oxygen atom is less in the DNA. Only one oxygen atom is less in the DNA. See, DNA, how many oxygen atoms? See, 1, 2, 3, 4. The 4 oxygen atom. Okay. In the ribose sugar, see how many oxygen atom? 1, 2, 3, 4, 5. So 5. So what is different we are seeing here? So the only the in between the two acids, only one oxygen atom is less in the DNA than the RNA. So this is about the chemistry of the nucleic acid. So next one. So how many the parts of the nucleotide? 3 parts. One is the pentose sugar, second one is the nitrogenous bases, and third one is the phosphate. In this part, we discussed about pentose sugar. So next one. In the next one, nitrogenous bases. Next one, nitrogenous bases. Nitrogenous bases. So in the nitrogenous bases, DNA and RNA have 4 bases. DNA as well as the RNA have 4 bases. See here, 4 bases are given. 1, 2, 3, 4. Next, first one. There is a 4 bases which we can take. One is purine, another one is a purine. How many purines are present? That means 2 purines are present. In one nitrogenous basis we are taking is 4 bases are present. We have remember properly. Nitrogenous bases are helpful for the formation of the nucleotide. These nitrogenous bases we are taking is there is a 4 type of the bases. The 4 type of the bases. So out of 4, 2 bases are purine bases. Two bases are called the purine bases. Another two bases are called the pyrimidine bases. First, we are taking the purine bases. The purine bases consist of adenine as well as the guanine. This one adenine as well as the guanine. These adenine and guanine are double carbon nitrogen ring. This one is one type of the nitrogen ring. Another one is the nitrogen ring. So the double carbon nitrogen ring is present in the adenine and the guanine. That means adenine and guanine have double carbon nitrogen ring substance. So it is called the purines. Next, thiamine, cytosine are the single ring carbon nitrogen. Single ring. So it is called the pyrimidine. So remember it. So there is a 4 bases in the nucleotide. That means nitrogenous bases are 4 bases. Out of 4 bases, 2 bases are purine and another 2 bases are pyrimidines. The purines we are taking means adenine and guanine have double carbon nitrogen ring substance. That is purine. Thiamine and cytosine have single ring called pyrimidines. So the uracil is present. That also single ring only. Uracil also single ring. So this uracil is present in the RNA. And other 4 bases are present in the DNA. Other 4 bases are present in the DNA as well as the RNA. DNA and RNA. See, adenine, guanine, cytosine, thiamine. These 4 bases are present in the DNA. Adenine, guanine, cytosine, instead of the thiamine, uracil is present in the RNA. Do you understand? Adenine, guanine, cytosine, thiamine. These 4 bases are present in the DNA. So, adenine, guanine, cytosine, and uracil. These bases are present in the RNA. These are the nitrogenous bases. Third part of the nucleotide. First part pentose sugar. Second part nucleotide, nitrogenous bases, and third part phosphate groups. See. Next one, phosphate group. See the phosphate group. Phosphate group derived from the phosphoric acid. Phosphate groups are derived from the phosphoric acid. The phosphoric acid symbol is H3PO4. It is a phosphoric acid. From which one the phosphate are derived? The phosphate are derived from the phosphoric acid. It has 3 active OH groups. This phosphoric acid has a 3 active OH group. Phosphoric acid has a 3. That one more question in the NEET exam. There is a 3 active OH group are present in the phosphoric acid. Okay. Out of these 3, 2 OH are involved in the strand formation. Out of 3, 2 OH are involved in the strand formation. So next, that also what? How many OH are involved in the strand formation? Like that they will ask the question. So for 2 OH are involved in the strand formation. How many OH groups are present in the phosphate, that is phosphoric acid? 3 OH groups are present. The bonds are formed from phosphates. The bonds are formed from phosphates. See, for example, adenine, guanine. For example, so that is connected by one. So these bonds are formed by the help of the phosphate. That is called the esters. 2 more questions. What is ester? The bonds are formed from the phosphate that are known as the esters. The oxygen atom of the phosphate group is negatively charged. That is a deep question. What is the charge of the oxygen atom? Whether negative charge or positive charge. The oxygen atom of the phosphate group is the negative charge. The negative charge phosphate ensure the retention of the nucleic acid. They ensure the retention of the nucleic acid within the cell or cell membrane. The phosphate ensure the retention of the nucleic acid within the cell or the cell membrane. So with the help of the phosphate only, it forms the nucleoside as well as the nucleotide. Do you understand? Phosphate is derived from the phosphoric acid. So how many OH groups are there? 3 OH groups are present in the phosphoric acid. So out of three, how many groups are helpful for the strand formation? Two OH groups are involved in the strand formation. So these bonds are formed from the, that is phosphate, are called the esters. The oxygen atom of the phosphate group is always negative charge. Okay, these are present in the cell as well as the cell membrane. Okay, so how the nucleoside and nucleotides are formed? You know about the pentose sugar, you know about the nitrogenous bases, and third about the phosphate. So with the help of these three things only, so the nucleoside and the nucleotide will form. So pentose sugar means you know DNA and RNA, deoxyribose and ribose sugar, only called the pentose sugar. And the second one is the nitrogenous bases. Nitrogenous bases we are taking means there is a purine as well as the pyrimidines. Adenine, guanine, cytosine, thymine, and uracil. These are the nitrogenous bases. Next, phosphate group. Phosphate group we are taking means so now three OH groups are present in the phosphoric acid. So these all the things are combined together and form the nucleotide as well as the nucleoside. Now see the nucleotide and nucleoside diagram. These are not given in your book now. So you want to know about what is the shape and size or arrangement of the nucleoside as well as the nucleotide. The nitrogenous bases link one molecule of the sugar forming is called the nucleoside. See here nucleoside, it's a ribose sugar. So with the plus the ribose sugar, only one nitrogenous base are attached. So this compound is known as the nucleoside. Do you understand? One pentose sugar, that is whether it is a deoxyribose or deoxy sugar or otherwise the ribose sugar. So combined with the only one nitrogenous bases. So there is a 5 kinds of the nitrogenous bases. Basically 2, purine as well as the pyrimidines. Purine we are taking is because the purines are the 2 ring form, 2 ring of the carbon form. So there is we studied about the adenine and the guanine. So adenine, guanine, cytosine, thymine. So in the DNA, adenine, guanine, cytosine, uracil in the RNA. So these nitrogenous, any one is attached with the pentose sugar that forming or giving the one of the substance that is known as the nucleoside. Pentose sugar plus one nitrogen base, so gives nucleoside. So remember nucleoside. Okay. So the nitrogen base linked to one molecule of sugar forming the nucleoside. Okay. Next, nucleotide. The nucleotide has a 3 part. One is the pentose sugar. Another one is the nitrogen base. Another one is the phosphate. That means we can say pentose sugar plus nitrogen base. So totally we are saying the nucleoside. Plus nucleoside plus one phosphate group form the nucleotide. Like that also we can say nucleotide. Do you understand what is difference between the nucleoside and nucleotide? So first we studied about the pentose sugar. So pentose sugar has a 2 types. One is the deoxy sugar, another one is the ribose sugar. So second one nitrogen bases. Nitrogen bases has a 2 types, purine and pyrimidine. Purine are the 2 bond carbon atoms, and pyrimidines are the single carbon atoms. That means single ring. Purine double ring. So it has a nitrogen bases, adenine, guanine, cytosine, thymine, and uracil. Okay. So next one, the third one is a phosphate group. Phosphate group has a OH group. Now these 3 also combine together to form the nucleoside and nucleotide. First, we want to know about how the nucleosides are formed. One pentose sugar plus one nitrogen base, whether it is adenine or guanine or cytosine or thymine or uracil, but only one nitrogen base combined with the pentose sugar, whether it is a deoxy or otherwise the ribose sugar. So it will form one of the substance that is known as the nucleoside. So with the nucleoside, the phosphate groups are attached and forming the substance that is called the nucleotide. That is known as the nucleotide. The phosphate group attached to the nucleoside forming the nucleotide. The question they will ask, differentiate between the nucleoside and nucleotide. Okay, the nucleotides join to form the polynucleotide chain. So, like this type of the nucleotide join together. See, one phosphate group, one pentose sugar, and only one nitrogen bases. But double ring only present for the adenine as well as what? Guanine. So that is a double ring nitrogen base adenine combined together and form the nucleotide. Like this type of the nucleotides continuously join together and form the one of the chainless structure that is called the polynucleotide chain. This hydroxyl group combined with the one nucleotide that only form the ester. We can say the ester also, we can pronounce that one. So one nucleotide combined with the one hydroxyl group. One hydroxyl group and form the ester. How the esters are forming? With the help of that means the one nucleotide combined with the one hydroxyl group and form the ester. Next, the chemical bonds with the sugar components with nucleotide are called phosphodiester bond. It is known as a phosphodiester bond. One word answer. The chemical bond with the sugar component with nucleotide. One nucleotide plus one sugar component, that is deoxyribose or ribose sugar, are called the phosphodiester bond. That only mentioning like a 5' or 3'. So this word is what? Prime. You want to pronounce it? Prime. 5' or 3'. This 5' or 3' indicate polarity of the strand. Polarity of strand. For example, DNA looks like this. This is the two strands. These two strands are mentioning 5' and 3'. This only knows the polarity of the strand. The two ends of the strand, that is the two ends of the DNA, designated by symbol 5' as well as the 3'. 5' as well as the 3'. What is 5'? 5' refers carbon in the sugar. 5' refers carbon in the sugar. And phosphate group is attached. The carbon only the phosphate is attached. See, the phosphate is attached in the one of the region. So that region is called pentose sugar. So the 5', this region is called a 5'. So in this section, pentose sugar, this part is called a 5'. See, this part only we are saying the 5'. Where the phosphate group is attached to the pentose sugar, that region is known as what? So 5' part in the sugar. Okay, 3. The 3 is referred the sugar which attaches the hydroxyl group. So the 3 is attached to the hydroxyl group. So this part is called that is a 3', 5' as well as the 3'. In RNA, we are taking this, the OH group is attached in the 2 position. OH group is attached in the 2 position. So that is in the 2 position. 5' as well as 3', direction of the nucleic acid is understanding for only replication and transcription. Replication as well as the transcription. One word this one. So remember it. 5' and 3' direction of the nucleic acid is understanding of replication and transcription. So this is about the phosphate group. Okay. So next one, sir. Now we are saying about the DNA structure. See, in the DNA structure we are seeing. So the DNA structure is model. The DNA model was proposed by Watson and Crick in 1953. One word more. Remember it. So the DNA model was proposed by Watson and Crick in 1953. Base pairs are present in between the two strands. See, this one is one strand. Another one is the strand. So the two strands are formed. This DNA has 2 strands. The 2 strands are present. The base pairs are present in between the 2 strands. See, base pair: adenine, thymine, cytosine and guanine. You already know nitrogenous bases are two. One is the purine, another one is the pyridines. Purine as a two, adenine and guanine. Pyridine we can say the cytosine and thymine. Whether RNA means the thymine, instead of thymine, uracil is present. Okay. So this is a DNA structure. So we can't see the uracil here. Okay. In the DNA structure, we can't see the uracil. Only we can see adenine, guanine, cytosine, and thymine. Okay. This for nitrogenous base are present in between the two strands. So the adenine always combined with the thymine. That means attached with the thymine. Adenine attached with the thymine. That also there is one of the bond, adenine and thymine. In between the adenine and thymine, one is a single bond. Adenine and thymine. That means we can see adenine and thymine attached like this, two hydrogen bond. See, adenine and thymine attached with the two hydrogen bond. Do you understand? The two bond is there. Guanine and cytosine attached with the three bond. Three hydrogen bond. So remember it. Adenine, thymine. Adenine always attached to the thymine. Guanine always attached to the cytosine. Adenine and thymine are attached by the two hydrogen bond. Guanine and cytosine are attached by the three hydrogen bond. That is the difference between these two nitrogenous bases. The ratio between the adenine and thymine and guanine and cytosine all are constant and equal. The only the two and the three hydrogen bond is present there, but we are taking the ratio. Means the ratio is constant as well as equal to the adenine, thymine, guanine, and as well as the cytosine. See this diagram. This also been given in your book. See DNA structure. The adenine, that is a D means deoxyribose. These are attached with the one of the phosphate as well as the nitrogenous bases. So it is attached to the nitrogenous base, adenine and thymine. And one side is attached to the phosphate. So it is called one of the nucleotide. Do you understand? One sugar, that is deoxyribose, plus one nitrogenous basis adenine. So we will give that. So that is called the nucleoside. That is known as the nucleoside. At the same time, deoxyribose plus one nitrogenous base, whether adenine or guanine or cytosine or uracil, plus one phosphate group, this side. It is known as the nucleotide. It is called the nucleotide. I think now you clearly understood this part. So the DNA we are taking this, that is a double helix model. It was first proposed by Watson and Crick in 1953. About the DNA, the next part we will read the elaborate. So for DNA structure, I am explaining this one more. So it has how many base pairs? 4 base pairs. So where it is present in the DNA, it is present in between the two strands. So adenine always combined with the thymine. Guanine always combined with the cytosine. So in between these two parts, two nitrogenous bases, 2 hydrogen bond and 3 hydrogen bonds are present. The ratio between the adenine and thymine and guanine and cytosine is always constant and equal. Okay. Next, RNA world. See, they will ask the question: give short notes on DNA or give short notes on nitrogenous bases. Okay, they will ask give short notes on nitrogenous bases like that. So what I given here, that one you want to write, read and write properly. I will send the PDF also for your reference. So each and every question there is a separately given the answer. Now next one we are saying about the RNA word. So one of the 10 mark question, that means 5 mark question, elaborate question in your exercise. Okay, so that is RNA word. But in your book they given like a paragraph. So we are changing the paragraph like a important keywords with the points. Okay, so that is a RNA word. In your book they given the different type of the diagram about RNA. So I given here a simple diagram about the RNA. Okay. Now this RNA is what? It is one type of the nucleic acid. ribonucleic acid. That means what type of the sugar is present? Ribo sugar is present there. What type of the nitrogenous bases are present in the RNA? Adenine, guanine, cytosine, and uracil. These are basic concepts. RNA means what? It is one of the nucleic acid. So what are the components are present in the RNA? One sugar molecule, that is called the ribo sugar. So it has a nitrogenous bases, that is adenine, guanine, cytosine, and uracil. Remember properly, uracil. Thymine suppose coming this, that is called the DNA. Uracil means that is a R. Okay, you can see in the diagram also. I say given adenine, uracil, guanine, and cytosine. Can you see that one? These four types of nitrogenous bases are present. Yeah. First, the cells contain 10 times as much RNA than the DNA. So, which one is more amount compared to DNA? The RNA is more amount than the DNA in the each cell. That also one need question. So, the cell contain two types of the nucleic acid, RNA as well as the DNA. But which one is the more amount means RNA? The RNA is more amount than the DNA. How much times it is more amount? 10 times more than the DNA. The Frankel and Contract and Singer in 1957 first demonstrated RNA. So the RNA was demonstrated by Frankel, Contract, and Singer. So it plays many roles in the cell. The RNA is playing many roles in the cell. The RNA is playing many roles in the cell. It is one of the genetic material. RNA is the genetic material. That's also very important. It is a genetic material in the virus. So we can say the example, tobacco mosaic virus. TNV virus means tobacco mosaic virus. Okay, the RNA is the genetic material, especially in the virus. The RNA word was first proposed by Leslie, Wargel, Francis Crick, and Carl Lewis. Already know about the Carl Lewis. We studied in the classification. So the RNA word was proposed by, it is one of the key questions. RNA word was proposed by Leslie, Wargel, Francis Crick, and Carl Lewis. The RNA is the first stage in the evolution of the life. The RNA is the first stage in the evolution of the life. The life is originated from the RNA. The RNA is the first stage in the evolution of the life. RNA is catalyzed all molecules necessary for the survival as well as the replication. One cell want to live in this world and it want to multiply. So that time, so we especially the cell need the RNA. The RNA only catalyzes all the molecules necessary for the survival as well as the replication. Survival means living. Replication is dividing. The term RNA word first used by, first I told you, proposed by. Now, the RNA word name was first used by Walter Gilbert in 1986. Walter Gilbert in 1986. In 1986, they believed RNA as the first genetic material under it. Already they told the RNA is helpful for the survival and replication. The RNA only the evolution of the life. So they believed RNA is the first genetic material under it. Now we can say, now everybody knows the DNA one has the genetic material. Before find out in the DNA is the genetic material or prove the DNA is genetic material. So everybody believed. So that is a first genetic material on the earth is the RNA. After Chase and Hershey and Chase experiment only, we concluded the DNA is the genetic material. The first before I thought about the Hershey and Chase experiment. In that from that experiment, we understood DNA only the genetic material. But in 1986, everybody believed RNA only genetic material on the earth. RNA is essential for the life process. So what are the life process? Like metabolism, translation, splicing, etc. Metabolism, translation, splicing, etc. Everything is takes place. So that is the help of the RNA. RNA acts as a both genetic material as well as a catalyst. So that act like a what genetic material as a catalyst. That means catalyst means it is increasing or it is supporting for any metabolism. Several biochemical reactions are catalyzed by RNA. So the RNA is known as ribosome. The ribosome is helpful for the protein synthesis. The Royal Blood Center we studied about the function of the RNA. The RNA is very, very important for the protein synthesis. So this RNA is catalyzing or making the reaction of the several biochemical reactions that takes place in our body with the help of the RNA. Its function like a gene regulation. In making the function like what? Gene regulation, making the regulation of the gene functions. And Andrew Fire and Claire Melo given the opinion about the RNA, it is an active ingredient in the chemistry of life. The RNA only giving the active part in the body. So it is called the active ingredient. Active ingredient of the body. Active ingredient in the chemistry of the life or the body. You understand? RNA word we are taking this. So totally compared to DNA, RNA is present more amount in our body. How many times it is more than means 10 times as much RNA than the DNA. So who was first demonstrated RNA means Frankel, Conrad and Singer in 1957. Okay, so the RNA is making many role in our body cell. So the RNA is the genetic material, especially for the many viruses. So example, so some viruses are in the DNA also genetic material. But so commonly we are saying this number different type of virus are in the RNA only as a genetic material. RNA word was first proposed by so the Bernasi, Wargel, Francis Crick and Carnus. But the term RNA word first used by Walter and Gilbert in 1986. Walter Gilbert in 1986. So it is one of the first stage in the evolution of the. It is a first stage of the evolution of the. So it is essential for the different type of process in our body, like metabolism, translation, splicing, etc. So the RNA acts as both genetic material as well as a catalyst. So because of the catalyst only, it is known as the called the ribosomes. So it functions like a gene regulator by binding to the DNA, and it's giving the opinion. It's one of the active ingredient in the chemistry of life. Active ingredient in the chemistry of life. So in the book question, there is one question. So you explain the RNA word. For that, you want to draw the RNA diagram, and you can write these points. That is enough for the answer. Okay. So next one, we are going to see about what is difference between the DNA and RNA. That means DNA wise RNA. DNA wise RNA. So in the DNA wise RNA, we are taking this first one. The DNA is present inside of the nucleus. We are taking this nucleus cell. So inside of the nucleus. Okay. Inside of the nucleus that is present the nucleic acid that contain the DNA. So RNA is present in cytoplasm. This is here. Only present in the inside of the nucleus. Inside of the nucleus. The RNA is present in the cytoplasm. Cytoplasm. RNA is present here as well as the nucleus. First point, see. Only DNA is present inside of the nucleus. RNA is present in the cytoplasm as well as the nucleus. Okay, next one. DNA is the genetic material. With the help of the Hershey Chase experiment, we conclude that the DNA is the genetic material. RNA is not a genetic material. It is not a genetic material. It is helpful for the protein synthesis. But in virus, it is a genetic. We are already told. Many viruses are having the genetic material RNA. So some viruses have the genetic material like the DNA. So many of the viruses are having the RNA is a genetic material. It is double stranded. It is a double stranded. Already you know about the structure of the DNA and RNA. So the structure of the DNA is double stranded, like this double stranded. RNA is the single stranded. RNA is the single stranded. Single stranded. RNA is the single stranded. So next third point, double standard, single standard. Next one, molecular weight. It is not given in your book, but it is important for the need one. So molecular weight, 3 to 4 million in prokaryotes. You know what is prokaryotes? Well-developed nucleus is absent, means that is called prokaryotes. That means the nucleus does not develop properly. Eukaryotes, well-developed nucleus is present, that cells are called eukaryotes. Do you understand? Well-developed nucleus we can see in the cell, means that is called eukaryotes. The nucleus does not develop properly, that is not well-developed nucleus is present. Only the nucleoid material is present. Nucleoid material is present. That is called the prokaryotes. Example, E. coli, bacteria. So, in the molecular weight we are taking is prokaryotes contain 3 to 4 million of the weight of the DNA. Eukaryotes have 263 million of the weight of the DNA. DNA weight is present 263 million in the eukaryotes. Molecular weight of the RNA 25,000 to 20 lakhs. 25,000 to 20 lakhs. Okay, DNA has a two types. One is intron and extron. So remember properly intron, extron. In the next part, you will read about the intron and extron in the protein synthesis. Okay, intron and extron. Exon, not extron, exon. Two type of the DNA. One is intron, another one is exon. RNA has three types. One is messenger RNA, another one is ribosomal RNA, transfer RNA. mRNA means messenger RNA. mRNA means messenger RNA. rRNA means rRNA, ribosomal RNA. tRNA, transfer RNA. tRNA means transfer RNA. So how many types of RNA? 3 types. How many types of the DNA? 2 types. Intron and exon. Next, DNA is known as a deoxyribose. RNA is known as a ribose. So deoxyribose is a pentose sugar only. Ribose sugar also what a pentose sugar. But only one oxygen atom is more in the RNA. One oxygen atom is less in the DNA. Remember that one. Nitrogenous bases, adenine, thymine, guanine, cytosine is present. Here, adenine, guanine, cytosine, uracil is present. Here, unusual bases. Here, modified bases. So, only that instead of thymine, uracil is present. It can replicate, DNA can replicate, but RNA do not replicate. DNA are long-lived one. RNA are short-lived, as well as what degrade easily. So only it is short-lived. Why RNA is living short-lived compared to DNA? Because it degrades easily. So only that's why it is very short compared to DNA. DNA is long-lived because it does not degrade very easily. So only it can live long time. Okay, these are the difference between the DNA and RNA. In your book, they will given the topic DNA wise RNA. Okay, so you can make note these points, remember these points. It is very helpful for your remarks. Okay, so next one. Next one, properties of DNA and RNA. Properties of the DNA and RNA. That was one of the 3-mark questions in here. Exercise question. So first one, next properties of the DNA or RNA. Properties of the DNA or RNA. So first one, self-replication. Second one, stability. Or third one, information storage. And the fourth one, variation through mutation. See, I given like a points here. You make a points and read it because it's one of the important theoretical questions. Write about the properties of the DNA and the RNA. First, self-replication. Both nucleic acids, that means DNA as well as the RNA. DNA and RNA have the ability to direct duplication. Both also have the ability to make a duplication. That means direct duplication. That is a self-replication. That means DNA also having the capacity and RNA also having the capacity to make it the replication. That is making the duplication. Stability. DNA is the, see already I told the DNA is having the replication capacity. RNA does not have the replication capacity, but it is given the ability to duplication. Don't get the confusion, replication and duplication. DNA has the capacity making the replication. RNA does not have the capacity making the duplication. Sorry, replication. But the duplication they are taking means both nucleic acids have the ability to give the what? Direct duplication. So remember properly. What is difference between the duplication and the replication? Okay, replication is absent in the RNA. Next, stability. DNA is having the more stability than the RNA. More stable and chemically less reactive when compared to the RNA. Remember, DNA is chemically more stable compared to the RNA. But real activity we are taking means, so the DNA has a very less activity than the RNA. DNA means it has a stable condition. Stable. But RNA does not have the more stable. Okay. But we are taking the reactive. That is activity condition. Reactive means, so chemically reactive is less, but here. So the DNA is chemically more stable and chemically less reactive when compared to the RNA. That you want to read like that. DNA has a more stable but less reactive. RNA has a less stable and more reactive. Less stable and more reactive. Okay. Presence of the thymine. So the DNA we are taking is adenine, guanine, cytosine, and thymine. So these are the nitrogenous bases. Adenine, guanine, cytosine, and thymine in the DNA. In the RNA we are taking is adenine, guanine, cytosine, and uracil. It is a RNA. Okay, so the presence of thymine instead of the uracil in the DNA confers additional stability to the DNA. The DNA having the more stable than the RNA. What is the reason? Because instead of the uracil, the thymine is present in the DNA. So only the DNA has a more stability than the RNA. So very important need question. Why the DNA is more stable than the RNA? Because the thymine nitrogenous base is present in the DNA, so it is more stable. The thymine is absent in the RNA, so but instead of the thymine, that is present uracil. So only the RNA has a less stable. So need question. So remember properly. Why the DNA has a more stable? Because of presence of thymine. Why the RNA has a less stable? Because of the presence of the uracil. You understand? So next one, information storage. So what is this? How the RNA and DNA are storing the information? DNA expressed in the form of Mendelian characters. Already studied the botany. Mendelian characters, recessive character, dominant character, so like that. These are expressing only in the help of the DNA. RNA directly helpful for the protein synthesis. That means code for the protein synthesis. Next chapter we will read about the protein synthesis. The RNA is very, very important for the protein synthesis. DNA depends on RNA. See, RNA directly helpful for the protein synthesis. The DNA also helpful for the protein synthesis, but it's not doing the function directly. So the DNA depending on the RNA for the synthesis of the protein. So it is also very important. RNA directly code for the protein synthesis. DNA does not or do not directly contact or directly doing the protein synthesis. So it only depending on the RNA for the synthesis of the protein. DNA and RNA both also genetic material, but the DNA is more stable at the storage of the genetic information, because the RNA is genetic material in the only the lower organism. That means especially we can say the virus. The DNA is a genetic material for the all the organisms. So the DNA is the most stable because of the presence of thymine. It is more stable and it making the storing the more amount of the genetic information compared to the RNA. RNA also transfer the genetic information, but it does not transfer more amount of the genetic information like a DNA because the DNA is more stable than the RNA. That also one of the new questions. The DNA only transferring the genetic material, so more amount compared to the RNA. So what is the reason? The DNA and RNA is the one of the genetic material in the viruses because the DNA is a more stable form because thymine is present. So the DNA is a more stable and may storing the more amount of genetic information. So compared to the RNA. In the RNA, instead of the thymine, uracil is present. So it is a very less stable in condition. Next one, variation through the mutation. Both able to mutate. Mutation means sudden change. DNA as well as the RNA can do the sudden change. It can make the mutation. RNA unstable. Why RNA is unstable? Because uracil is present, so it is unstable and very mutate very fast. It is unstable, so it is easily making the mutation. Mutation is sudden change, but RNA virus has short life. So you know about the coronavirus. Very easily that is what multiplying, changing the genetic characters. And then we hear in the news also, because the RNA is unstable in condition. Why it is unstable? Because the nitrogenous bases are very weak because uracil is present there. So only it is easily mutate together. So only we are seeing nowadays coronavirus, alpha, beta, delta, like that. Very easily it is changing their activities according to the host body. So that is a reason. But DNA can't do like that. Okay, because it has a very strong stable condition. So it can mutate and evolve faster. RNA will make the mutation very faster. So only we can make the structure and shape, everything activity are very fastly. So changing according to the host body. These are the properties of the DNA as well as the RNA. Do you understand? Do you understand? Shall I ask the question?